All right. I went ahead and started recording. It's really early. It's 30 minutes until class starts. But just to make sure I don't forget to start recording when the lecture starts, and just to make sure there's no technical difficulties later, I'm gonna go ahead and start now. So, if you're watching this video, notice on the screen it looks really blank, right? But if I zoom out, then you can see everything, right? So, what you can do is keep an eye on your screen. Fast forward, keep an eye on your screen until you see where it's zoomed out, and then you'll know that we're saying some important stuff, and you'll need to start paying attention. Of course, that might be before the actual lecture starts, because as you probably know, if you're somebody who watches the videos often, there are often announcements before we get started. So anyway, that's that. Just keep an eye on, like I said, fast forward. You need to probably go about, I don't know, 20, 25 minutes forward. And like I said, just keep an eye on the screen. Look forward to zoom out, and that's when you'll know to stop going fast forward. And while I'm at it, of course, hopefully, I would assume nobody's listening right now because you're probably fast forwarding, which is okay. But just in case, um, I'm going to be perusing through some biology videos online to see which ones might be good to share with, with, the students. So you might hear that in the background. So I assume if you followed my earlier directions and were fast forwarding until you saw this change on the screen, that you probably saw this change on the screen and you stopped fast forwarding. So sorry for the false alarm. I am just looking at some biology videos, and that's why I'm presenting the screen. So you can watch these little videos if you want. Otherwise, like I said, just keep an eye on the screen, and you'll see once this other video stops. And again, once the camera zooms out, then you'll know when it's time to stop. Maybe I'll even wave at the camera. That's yeah. If I could remember, I'll wave at the camera too. Again, as a sign to stop fast forwarding if you want and start watching. Once again, I'm just reminding you if you happen to be fast forwarding and if you just stopped recently, you might be like, hey, what's going on? You can just keep fast forwarding. And just in case you are actually watching this, let me back it up here a little bit. He talks about a Rube Goldberg machine. Might as well show it. Goldberg machine. Uh, yeah. It's yeah. So you've seen him probably your whole life, but maybe never heard it called that. Look, start something off. In this case, it looks like a truck hitting dominoes. Here we go. Anyway, if for some reason you're watching this instead of fast forwarding it, let's give you an extra attendance word so you can get a little extra credit. Um, so this first hidden extra credit word is golf club, since we just saw one. So golf club is the hidden extra credit word. So when you send the list of words, you might want to also indicate maybe in parentheses like this is extra credit, you know. So golf club parentheses extra credit. And maybe I'll have some more between now and when class starts, but we'll see. Anyway, that's a Goldberg machine. That's what they're referring to in the other video. An interesting analogy comparing that, like he just said, you need ATP to start this process, and the whole purpose of this process is to make ATP, which might sound counterintuitive, right? Like, why do we start the process with ATP, but we're trying to produce ATP? One way you could look at it, like on a bigger scale, is when you think about energy, right? Not cellular energy, but for example, electricity, right? We need electricity. Morning. Pretty excited. Too excited for something so small. So I'm sure you've seen the videos on the like in the pre-labs and stuff, the crash course videos. They have new ones. I'm excited. Now, don't get me wrong. I love Hank Green, and I have a feeling I'll probably like his better. But the fact that there's new ones kind of excites me. So they're only 4 weeks old. This one's only 4 weeks old. So yeah, I'll have to check this out later. I like it though. When I was a student, I was trying to learn something that was particularly difficult for me. Usually, the best thing to do was to learn it all kinds of different ways. I talked to the tutor and see how they explained it. Talk to the professor, see how they explained it. Talk to my classmates who knew what they were talking about, see how they explained it. Looked it up on, looked up videos. So it really helps. Everybody's brain works different ways, so it's good to find the way that works for you. Morning. Let me zoom this out. There we go. All right. Burton is online. Fisher is online. Let's see. And just for anybody online or here, again, extra credit. I'll just throw in another word. Let's see, golf ball. Not much, but it'll help you since you guys are here earlier. That means if you want to, when you send those words in, you'll be getting more extra credit than people who get here on time. Not by much, but whatever. Every little bit helps, right? So golf ball is the extra credit word. The extra credit word, if you will. Andrea is online. And we are already recording. Not that you guys ever say anything that shouldn't be recorded, but just in case, we are being recorded. All right, let's see. Okay. So since we're recording, you guys don't have to pay attention if you don't want. I don't care. But since we're recording, I'll just do more reminders and announcements. Let's see. I don't think there's anything new. It's just old stuff at this point. You can retake the first exam now. You can retake it as many times as you want. The retake is open book. It's not timed. What else to tell you? You have to let me know when you're done. Otherwise, your score won't automatically adjust. You have to tell me because I need to go in and average your first score and your last score. So make sure you let me know when you're done with that so I can adjust your score. If by some weird miracle you keep retaking it, but when it's not timed and you have an open book and you somehow keep getting a lower score, then we'll just keep your original higher score. So you have nothing to lose. Also, I'm going to keep that new exam one open because half of the final exam, remember, it's about 100 questions. Half of it's going to come from the first 3 exams. Verbatim, I'm going to take questions, copy and paste them into the final exam. So, it would be helpful to have that as a study guide. What else? You should have 40 points of independent work by now. As you've noticed, it keeps going down, how much it's worth. The syllabus says it's worth 10 points, but then like day one, I said for now it's worth 20 points and it'll keep dropping. So take advantage of it. It's worth 15 points right now and it's going to drop again. So good. Yes. Definitely when I grade it. Yeah. Yeah. Good question. Because otherwise we don't know how much it's worth, right? Until you could turn in 10 pages and And then after I go through it and do my adjustments, it might be worth, I mean, this is an exaggeration, but your 10 pages might only be 1 page worth of graded material. A great question. Yeah. But yeah, like I keep saying, make sure you turn it in. This is mostly for the people who haven't turned anything in yet. Because for those of you who have turned something in and have had me grade it, you see how I grade. You see how I change the paragraphs and how I delete these things and how I question you on those things, right? And that's okay because you guys have plenty of time to do it. But I don't know who, but there's got to be at least somebody in this room who's going to wait until the due date to try to turn it all in. And it won't work out. Well, maybe it will. Who knows? But historically, never have I had a student wait until the end to turn in their independent work and turn it in perfectly. There was always some kind of mistake that they weren't aware of because they've never turned in an independent work paper before. So make sure you do that. What are some other reminders? That's not much. Not much. Again, because this is a big one. I know I keep saying it a lot. But speaking of independent work and people not doing it until the end, someone's going to ask this question on the very last weekend, even though I've been saying it all weekend or all semester. There is no minimum. You can turn in a 2-sentence page or paper. Just keep turning stuff in. It doesn't have to be a long paper. Well, it's 7:58. I'll start officially doing attendance now. Abigail is here. If you're wearing WVSU gear and I don't notice, make sure you let me know. Or if I say your name, say Go State or something. I don't know. Kincaid is here. Andrea is online. Mullins? No Mullins yet? Lewis is here. Cooper? No Cooper? Hunter is here. In WVSU gear? No. Oh, yes. Okay. Both Meltons are here. Crowder is here. Reinhardt is here. Like I said, both Meltons are here in person. Julia, Rogers, Talbert, Kennedy is here. Hannah is online. Larissa is here. Madeline, not yet. Hughes, not yet. Galloway, not yet. Cook is here. Durgan is online. Tate is here, right? Yep. Tate is here. Burton is online. Kinder. No Kinder yet. Jones. Canterbury is here. Tristan. Hobson. Ray. And Fisher is online. It's 8 o'clock. So did I miss anybody? Wait till 8:01 before we get started. Julia is here. Oh, I know a good reminder. Or maybe announcement. This might be the first time I've said this. No, it's not. It's not the first time. When you're doing the labs, you really should, if you can, walk around and talk to your classmates, right? You might be doing experiment number 1, but figure out what's going on with the people doing experiment 2 and 3, especially last week. I don't know if it was Monday or Wednesday section. The one where, which one was it? Oh, yeah, the one where you weighed one of the sucrose or the starch one, either one. There was some really weird data. And some people have lost points because the weird data, probably they didn't do the experiment and they're trying to answer the questions based on this weird data. They probably have no idea what the hell happened because I don't know what happened by looking at it. So look at that stuff before you leave. Look at it and try to answer the questions, at least in your head, before you leave. And be like, oh, I better ask them about this. I might have to answer a question about this. Let me talk to those people who did that experiment because this makes no sense to me. Does anybody remember what I'm talking about? The weird experiment from last week? Anybody remember? I don't expect you to because I don't even remember, and I just graded it yesterday. But anyway, the data was wonky. What was it? Something was gaining right. So anyway, it doesn't matter. That's also a good time to remind you, if I make a mistake on your grading or you think you got something right, tell me. I don't consider that an argument. Maybe I just misunderstood what you were trying to say. So let me know, and maybe we'll fix your grade, or maybe you were wrong, and it'll be a good learning experience. And then you'll learn from it. And I bet, like I said, a situation like that, you're probably more likely to learn it. Anyway, any questions about anything? It's officially time to get started. I see Kinder is online. Let me write that down, then we'll jump back into it. There we go. All right. We're all accounted for. I was hoping some people would come in before we got started, but here we go. Back to this, cellular respiration. What we finished before we left on Monday was the overview, right? This was one of the last slides we saw because we just did the whole overview of the entire process of cellular respiration, from glycolysis to citric acid to electron transport chain. This picture is not in your textbook. It's from your old textbook, but it's a great picture to study because there's so many test questions on here. And I know I already said this, but I'll say it again really quickly before we get started and start going into details with each of these 3 steps. But remember, a lot of this test, especially for the cellular respiration part, is going to be me saying, in which stage does this happen? In which stage is this produced? In which stage is this used? So just looking at this one picture, which stage happens outside of the mitochondria? Glycolysis. Which stage happens inside the mitochondria? Citric acid cycle and electron transport chain. Which stage uses glucose? Glycolysis. Which stages produces pyruvic acid or pyruvate? We call it pyruvate. I'll tell you why later. Glycolysis. Which stage produces ATP? All 3 of them. Which stage produces the most ATP? Electron transport chain. Which stages strip away high energy electrons? Glycolysis and citric acid cycle. And which stage uses high energy electrons? Right here. They all get stripped away and go to the electron transport chain, which is all in the name, right? Anyway, a lot of test questions right there. So let's move forward with that. Actually, before we move forward, let's do attendance words. I guess the first word will be acid. The first official attendance word will be acid. Anyway, with the big picture out of the mind, or out of the way, let's jump into details. Now we're back on track with your textbook. Because remember, what we just talked about, the overview, your textbook doesn't do an overview. So I did the basic overview, and now we're going to dig into more detail. So during glycolysis, the first thing that happens, well, sort of, is that we have a 6-carbon glucose molecule. Of course, all glucose, by definition, has 6 molecules, but that's just sort of a reminder that this glucose has 6 carbons, right? It's split in half. So when you split something with 6 carbons in half, you're going to have 2 things, right? If you split anything with half, you're going to end up with 2 things. And in this case, since the original thing had 6 carbons, then the 2 things that you're going to form have 3 carbons apiece. Does that make sense? It's a 6-carbon molecule. You split it in half. Now you have 2 3-carbon molecules. Make sense? That's the first step. More importantly, and I shouldn't say more importantly, to make things a little bit easier for you, for the exam, what you need to know is that during glycolysis, glucose is used, pyruvate is formed. It's that simple. I'm not going to ask you how many carbons glucose has, though at this point you probably know C6H12O6. You've seen it a lot. I'm not going to ask you how many pyruvate are formed. I'm not going to ask you how many carbons each pyruvate is made of. What you need to know for the exam is that we start with glucose and glycolysis, and we end with pyruvate. There might be some extra credit questions where I go in that deep, like how many carbons of glucose are there? How many molecules of pyruvate are formed? How many carbons are in pyruvate? So I might do that for extra credit, but I'm definitely not going to require it for regular credit. So the important thing here, not for the exam, but for this process, what's the most important thing here is we have high energy electrons stripped away. Because like I already said with that whole overview, like the whole thing's about making ATP, right? And the electron transport chain makes the most ATP by far of any of the 3 stages. So in a sense, the first 2 stages are basically there to service the 3rd stage. It's all about that 3rd stage. And what does the third stage need? As the name indicates, it needs high energy electrons. So where do we get those high energy electrons? From the first two stages. It's all about stripping away these high energy electrons. And it's a good time for me to remind you this. Yes, technically NAD plus is different than NADH. But as far as we're concerned, this conversation we're having, you could think of them as the same thing. And I'm not going to test you on the difference between the two, but really quickly, just to remind you, whenever you see any molecule that looks like that, for this exam, you should think that is something that's an electron carrier. It's moving the electrons from one place to the other. So that's exactly what you should. There's other ones too that you're going to learn about later. FADH2, there's some that we learn about in photosynthesis. They all look similar. So no matter which one you see on the exam, you should see that and think, yeah, that's the thing that's carrying electrons. But anyway, we're stripping away the high energy electrons. And technically, like I said, when it's NAD plus, you can think of it like as an empty bucket. That's the bucket that carries electrons. It's empty. And then when you put the electrons in, then technically it's called NADH, right? But once again, you're not going to need to differentiate between the two. You should just think of something like that. That molecule is the electron carrier. Another thing you don't necessarily need to know is this particular number. Yes, in glycolysis, we net 2 ATP. But what you need to know for the exam is not the specific number 2. You just need to know that glycolysis produces a little bit of ATP. Citric acid cycle, the one we're talking about next, produces a little ATP. The big one produces a lot of ATP, and it's specifically 32. That's the number you're going to need to know. For this, just know that it only produces a little bit. The entire stage is broken down into 2 phases that we're going to talk about. I'm going to put a big question mark next to this. Let's see if this thing works. No, that would be too easy. Anyway, big question mark. I'm not sure if I'm going to ask you if I'm going to go into this much detail or not. So that's why there's a question mark. Maybe I'll ask you about the 2 phases. Because here we go, big picture again. To remind you, cellular respiration itself is broken down into 3 stages. We're talking about the first stage, and that first stage itself is also broken down into 2 separate phases. So again, maybe I'll ask you, maybe I won't. Go ahead and study it. At the very least, maybe there'll be some extra credit questions. But anyway, yes, the 2 phases are energy investment and energy harvest. So the next slide is going to show what I'm talking about, and then after that, I'll take some questions. Actually, no, not yet. Are there any questions about this slide? All right. Again, a picture of what we're talking about. We are talking about glycolysis, which produces a little bit of ATP. It happens outside of the citric acid cycle. Again, the input, a 6-carbon glucose. The output, 2 3-carbon pyruvates. And again, to remind you, yes, glucose has 6 carbons. Yes, pyruvate has 3 carbons. No, you don't need to know that for the exam. Yes, you start with 1 glucose and end up with 2 pyruvates. Again, those numbers aren't important. The important thing for the exam is you start with glucose, you end with pyruvate. And again, I'll remind you, depending on your source, what you're reading, sometimes it's called pyruvate, sometimes it's called pyruvic acid. Those 2 are the same things. I call it pyruvate on purpose. You'll see why later. I do it to make things easier for you. Here we go. Like I said, this whole thing is broken down into 2 phases. Here's the first phase, energy investment phase. It's interesting, right? But the whole idea here is to get energy. We're trying to make ATP. Yet to get the whole thing started, we need ATP, which would be a good independent work topic if you really want to dig into something deep. Well, I guess you probably wouldn't have to dig into it. I bet you could Google it and find one quick, simple answer. But anyway, the question that you could look up if you want is, if it takes ATP to start the process of cellular respiration, and if cells get cellular ATP from cellular respiration, then how did the first cellular respiration happen? Where did that first ATP come from? If you have to have ATP to start the process to make ATP, where did the first ATP come from? Something to look into for independent work if you want. You don't need to know it for this course. Anyway, this is the energy investment phase. You know, we take 2 ATP, we use those, and that's what we need to break up that glucose into the 2 pyruvates. And you can see, not that it matters for the exam, actually it doesn't matter at all, but anyway, you can see that the phosphate groups from those ATPs were transferred, just in case you're wondering. So it's not important to the story, but if I was looking at this and had no idea what was going on, I'd be like, where do those P's come from? So just to remind you, the P's came from the ATPs. And they're not important for the story. at least the way we teach it, because we're only skimming the surface. Anyway, next comes the energy harvest phase, right? So energy investment phase, if I were to ask a question about it, which I probably won't, in the energy investment phase, we use ATP to break up glucose and produce pyruvate. So technically, it is the energy investment phase that gives us the pyruvate in it. So right, that if there was a test question, that would be it. Which of the two phases of glycolysis produce the pyruvate? Energy investment or energy harvest? And the answer is energy investment. Likewise, which of the two phases of glycolysis use glucose? And the answer is energy investment phase. And then finally, after we've broken down the pyruvate, then some more stuff happens. We don't talk about what in this class. It's pretty complicated. There's a series of chemical reactions. But this is the part where we're finally stripping away those high energy electrons. Hence the energy investment part, right? Here, we haven't created any, or we don't create energy anyway, as we know from thermodynamics. But here, no energy was released, right? We just broke something. Here, we're finally starting to harvest that energy because we're stripping away those high energy electrons. And yes, as you can see for the picture, not that it's important to understanding what's happening, but just so you know, yes, another phosphate was added. Right, so here, this one only has one phosphate, right? This thing has two phosphates. Those things have names. You don't need to know them. And you don't even need to know that they have phosphates. I'm only pointing it out just because if you're studying this and you're like, where do those come from? And is that important? And I'm telling you, no. For us, that is not important. What is important, again, and I've already said it, I'll focus on it again. What we are doing here is stripping away high energy electrons. That's what it's all about. This whole thing, all of cellular respiration is about making ATP, and it's the third stage, electron transport chain, that makes the most ATP. And the first two stages are basically here to support the third stage by stripping away high energy electrons. So as far as what the exam is concerned and this story, that's the most important thing. Any questions so far? All right. So now, finally, some other things happen. And I like how there's multiple arrows. So that's a chemical reaction, that's a chemical reaction, that's a chemical reaction. There's 4 different chemical reactions that we don't talk about in this course. But between those 4 chemical reactions, we strip away some more ATP, or we make some ATP. And you don't even need to know how. You can look it up if you want. But notice I didn't tell you how we make ATP in glycolysis. All I'm telling you is that we make a little bit of ATP in glycolysis, which is a hint, not a hint necessarily, but I'll tell you this. When we get to the third stage, as you might imagine, I'm going to say, look, we make ATP. And for the third stage, I'm going to tell you specifically how we do it. And on the exam, there's going to be a question, which of the 3 stages uses this method to make ATP? Well, the answer is the third stage. That's the only one where I'm telling you how it's made. So I'm not telling you how it's made here on purpose. But anyway, look, we spent 2 ATP. We made 4 ATP, which means we netted 2 ATP. Does that make sense? Spent 2, made 4, so that means our profit was 2. But once again, the numbers don't matter. I'm just pointing that out. What matters is that we made a little bit of ATP, and we stripped away some high energy electrons. Any questions about that? So this is from a picture from your old textbook, and I need to refresh my memory. I don't remember what your new textbook says, but like I said, I don't get specific on how the ATP is made in this first stage, but here's a picture. I'll just let you know, and I'm not getting specific. I'm not giving that a name. I'm not going to test you on this, just in case you were curious. It's done by an enzyme. There's an enzyme that takes a phosphate from this molecule and puts it on ATP. You don't need to know that, but again, I just wanted to throw it out there just in case you wanted to know. And you could look it up for independent work if you want. What is the name of that enzyme that does this? What is the name of this molecule that gives away one of its phosphates? Once it gives away one of its phosphates, what is the name of that molecule, right? It's a different molecule once you start taking parts off. And what is the name of that process? That chemical reaction has a name. You can look it up if you want for independent work. Enzyme is your next word for attendance or participation. All right, so that's it for stage 1. Are there any questions about stage 1 before we jump into stage 2? All right. The next stage is citric acid cycle. Oh yeah, let's do the learning objectives first. By the end of this section, you will be able to describe the location of the citric acid cycle and oxidative phosphorylation, which again, to remind you, to make things a lot easier for you, I call that the ETC, the electron transport chain. You should be able to describe the overall outcome of the citric acid cycle and oxidative phosphorylation, again, electron transport chain, in the terms of the products of each, meaning and this is straight from your textbook. I didn't write this. That's a textbook author way of saying you should be able to tell me what the products of each are, right? They try to sound fancy. It's pretty simple. And then finally, you should be able to describe the relationship of glycolysis, the citric acid cycle, and oxidative phosphorylation, aka ETC, in terms of their inputs and outputs. Again, just a fancy way of saying what I've already told you. You need to know the inputs of the 3 stages and the outputs. And we've already talked about the first one, right? The input for the first one is glycolysis. The output is pyruvate and a little bit of ATP. That being said, let's move forward. So, like I said, the end product of glycolysis is pyruvate. So pyruvate, you would think, would go into the citric acid cycle. And that's sort of true, but not quite. Because this pyruvate is not ready as is. That molecule cannot go into the citric acid cycle. So it has to be groomed or changed or whatever adjective you want to use, modified. It's not ready to go. So let's talk about this. What happens is, one of the things we do to this pyruvate, not that you need to know, but to remind you, pyruvate has 3 carbons. So here we're lobbing one off. So boom, right away we've created CO2, and now we have a 2-carbon molecule. So I'll try to draw this. It probably won't work. 1, 2, 3. There's our pyruvate, right? Then we lob one of those carbons off. Now we have a 2-carbon molecule, and of course the 3rd carbon as CO2. So again, actually, let me say this really quickly. What I will not ask you on the exam is specifically how the CO2 was created, because that could be a test question. I could say, how was CO2 created? You could say CO2 was created in the first part of citric acid cycle when a carbon was removed from the pyruvate, but you don't need to do that. The test question is going to say, in which of the 3 stages was carbon dioxide produced? And the answer is citric acid cycle. I'm keeping it simple. I mean, maybe some extra credit questions where I do get that specific, but you don't need to know that. So again, we had a 3-carbon molecule. We lobbed one of the carbons off to make carbon dioxide. Now we're left with a 2-carbon molecule. That thing has a name. It's called acetic acid. You don't need to know the name of that thing either. But since I'm teaching out of the textbook, I'm mentioning it. That way, when you're studying the textbook and you're reading it, you're not like, whoa, I don't remember him saying this. So there you go. I'm saying it. The little star is to tell you, or to remind me to tell you, I'm not going to ask you anything about acetic acid. If I did, the question would be like this. Which of the 3 stages of cellular respiration is associated with acetic acid? The answer is the citric acid cycle. And here's a little hint for you. When we're talking about respiration, if there's any question that asks anything about acid, then the answer is citric acid cycle. So hopefully that'll be easy for you to remember, right? Citric acid cycle is associated, acetic acid is associated with citric acid cycle. So that's why it's the throwback. Remember I said earlier, pyruvate is also called pyruvic acid. That's why I don't call it pyruvic acid. Because in this exam, if you see any question that has anything to do with acid, the answer is citric acid cycle. You guys get it? That's why, to make it easier. And also to remind you, the citric acid cycle is also called the Krebs cycle. But once again, to make it easier for you, that's why I don't want to call it the Krebs cycle, because I want you to hear that acid, citric acid cycle, and that'll help you remember. If it's an acid question, the answer is citric acid cycle. Anyway, so again, we had pyruvate, we lobbed the carbon off, now we have something called acetic acid. That still cannot go into the citric acid cycle, right? So we're still in that in-between zone between glycolysis and the citric acid cycle, right? We're still grooming that pyruvate. So here we are at acetic acid. We need to make some more changes. So in this process, I got ahead of myself. In this process of lobbing this carbon off to make acetic acid, We also strip away high energy electrons, which again is the most important thing as far as this story is concerned, right? The first two stages, glycolysis and citric acid cycle, it's all about stripping away those high energy, those high energy electrons that will get used in the last stage, which is electron transportation. So there you go. And again, not that I have to keep repeating this because this is going to be the same thing over and over. This is the molecule, NAD plus, NADH. That's the molecule that's carrying the electrons from one place to the other. And technically, again, just to remind you, not that I'm going to test you, NAD plus is the name of the molecule before it gets the electrons, and then after it gets the electrons, it's technically called NADH. Each acetic acid that's produced is then attached to a molecule called coenzyme A to form something called acetyl-CoA. So it's the acetyl-CoA that then well, I guess you could say it differently. It's that CoA, that coenzyme. Remember we learned about coenzymes the other day? We talked about coenzymes and vitamins and things like that. Well, here's an example of a coenzyme. That coenzyme escorts the acetic acid into the first reaction of the citric acid cycle in the mitochondria. So finally, we got to the point where we can actually start the citric acid cycle. Big picture, really quickly to say it again. We started with 2 pyruvates, right? Each one of them is a 3-carbon molecule. We lob one of those carbons off to produce carbon dioxide and something called acetic acid. That acetic acid still then needs to be modified. So we attach it to something called acetyl-CoA. Coenzyme A, which gives us acetyl-CoA, which sounds really confusing. So let's take another look at it, but let's talk about what you need to know for the exam. For the exam, you need to know we produce carbon dioxide in the citric acid cycle. Acetic acid, or any acid, is always associated with the citric acid cycle. NADH, that's the thing that's moving electrons from one place to the next. And which of the 3 stages is associated with coenzyme A or acetyl-CoA, either one, and that's associated with citric acid cycle. So if you're making flashcards, it's just enough to know that coenzyme A and acetyl-CoA are associated with citric acid cycle. I'm not going to test you so deep where you need to know, oh yeah, well what happens is coenzyme A attaches to the acetic acid, and that gives us acetyl-CoA. You don't need to know all that. As long as you can associate acetic acid and coenzyme A and acetyl-CoA with the citric acid cycle, you're fine. Maybe I would ask you an extra credit question and say, how are acetic acid, coenzyme A, and acetyl-CoA related? And the answer to that question will be, well, acetic acid, when you attach it to coenzyme A, you get acetyl-CoA. But again, not going to go that deep for this exam. So your 100 level non-biology major course. Any questions about this slide before we talk about the actual citric acid cycle? Now that we have our acetyl-CoA, we're ready to get inside the mitochondria. All right. So again, citric acid cycle is the next step. That's what we're talking about. And this is the first stage that actually happens inside the mitochondria. So this picture is from your old textbook, but it's a good overview. Again, everything we just talked about, this is a picture of it instead of that stupid picture I tried to draw. Here we have pyruvic acid or pyruvate. I should fix this to say pyruvate. But anyway, yeah. Technically, we actually have 2 of these coming in. But anyway, we have pyruvate coming in. Again, we lose a carbon from that 3-carbon molecule, which gives us acetic acid. We attach the acetic acid to the coenzyme A to form something called acetyl-CoA. In the process, most importantly, this is what we're after here, is we're stripping away high energy electrons because that's what's needed at the end. We need all those high energy electrons. So any questions about this picture, which is just a visualization of the previous slide? All right. So again, now we're finally talking about the actual citric acid cycle. Right now, this acetyl-CoA, it's inside the mitochondria. It's starting the process, right? Overall, the acetic acid is dismantled down again to carbon dioxide again. Right? Because remember, acetic acid is a 2-carbon molecule. So overall, what's going to happen is it's going to be broken down into carbon dioxide again. But once again, you don't need to know that much detail for the exam. That portion, what you need to know for the exam is that CO2 is produced in the citric acid cycle. And we've already said that, right? Even in that little last stage, when we turned pyruvate into acetic acid, we made carbon dioxide. But here we are, we're making it again. Specifically, two molecules of carbon dioxide. Not that you need to know that number. Again, carbon dioxide is produced in the citric acid cycle. That's what you need to know for the exam. Your book does also remind us that some ATP is produced directly from this process, right? So yes, just like glycolysis, the citric acid cycle does produce a little bit of ATP. But more importantly, like I keep saying, here it is again in writing, what we're really after is high energy electrons, right? We're after those high energy electrons. We're trying to strip away high energy electrons. Remember I told you earlier, whenever you see any molecule that looks like this, NADH, you should think, oh yeah, that thing's carrying electrons. Well, here's the other one. Then there's something called FADH2. As far as we're concerned, those are the same thing. They do the same thing. There is a difference, but we don't need to know it. When you see a molecule that looks like this, and again, there's going to be a third different one in photosynthesis, but it doesn't matter. It's going to look similar. In this exam, when you see anything that looks like that, you should automatically think, oh, that's the thing carrying electrons. And I'll remind you, I'm not going to ask you the differences between NADH or FADH2 or the other one that I'm going to teach you later. You just need to, again, associate that with electron carriers. Anyway, another thing to point out is that every single carbon atom that enters the cycle as a fuel is accounted for as a carbon dioxide. But then there was a 4-carbon acceptor molecule that's recycled. And I will put an X through this because I'm not going to test you with that much detail. But again, teach out of the textbook. I wanted to say this. That way when you're studying your textbook, as I'm sure you guys do every night when you're reading your textbook, when you read this part, I don't want you thinking, no, he never said that. Well, I'm telling you now, right? Again, the important thing in this bullet point is the same thing I already said earlier. Carbon dioxide is produced. But yes, technically, just so I can explain it, hold on, let me see if it's on the next slide in the picture. Yeah, I'll talk about it with the picture. So are there any questions about the words before I show you the picture of these words? All right, so again, here's what we have. We start with this 2-carbon acetic acid, right? And it attaches to this 4-carbon acceptor molecule right here. And that forms, boom, the 6-carbon citric acid, right? Citric acid molecule. And then that thing goes through the cycle. We lob off 2 more carbon dioxide. There's 2 more carbons to form carbon dioxide. 6 minus 2 equals 4. We're back at the acceptor molecule. The whole process starts again. Granted, this is a really, really, really simplified version. There's a lot of different molecules between here and here. And there's a lot of different molecules between here and here. And they all have names and all their, they all have a specific enzyme that does those, does the chemical reaction. But we don't talk about that. The important thing here for the exam is, let's look at the exam questions. Which of the 3 stages is associated with acetic acid? Citric acid cycle. Which of the 3 stages is associated with citric acid? Obviously the citric acid cycle. If anybody misses that, I'm going to cry. Which of the 3 stages strips away high energy electrons? Well, citric acid cycle and the previous one, glycolysis. Which of the 3 stages produces ATP? All 3. Which of the 3 stages produces carbon dioxide? Citric acid cycle. Yeah, so that's all the test questions on this. And again, this is a simplified version. You start with this 4-carbon molecule, you add 2 carbons onto it, you knock one off to form carbon dioxide, you knock another one off to form carbon dioxide, you back up the 4-carbon molecule, and the cycle continues. Anyway, are there any questions about this slide? All right, pretty simple, right? All right, before we talk about the last stage, which is electron transport chain, or as your book calls it, oxidative phosphorylation. That's more complicated. Have to make it complicated. Let's talk about some big overall concepts before we jump into it. During the entire process, all 3 stages of cellular respiration, electrons fall, so to speak, in a cascade down an energy staircase, which is a way of saying when we start this process, those are really high energy electrons that are in that glucose molecule. And as we strip them away and then bring them to the electron transport chain, each stage, they're kind of losing that energy because it's a really unstable state. They can't stay high energy. So they basically lose that energy as they go from one place to the next. Which brings us to this bullet point. Electrons have to go somewhere. Just like in an electrical circuit, just like plugging something in or jumping your vehicle, electrons have to come from somewhere and they have to go somewhere. And this will definitely be a few test questions. Where do the electrons come from in this whole entire process? And I've already told you. I want to see if anybody can. I just haven't worded it in this way in the form of a question. So let's see if anybody can answer. Where do the electrons come from in this process of cellular respiration? Which molecule? That's a better way of putting it. What molecule provides those electrons? That's all right. The answer is glucose. You need to know that for the exam. Glucose is providing those high energy electrons. We strip them away in glycolysis. Then technically we strip them away from pyruvate, but that was still ultimately that was glucose to begin with. And we strip them away from citric acid, and we strip them away from acetic acid, right? All that stuff. So the answer to that question is where the electrons come from? They come from glucose. What molecule do they go to? Well, I'll go ahead and tell you now. Well, we'll talk about it in more detail when we get there. But they come from the glucose, and they go to oxygen. You need to know that for the exam. This is all, again, this is a lot like a circuit. So when you're jumping a car, the electrons are coming from the vehicle that has a not dead battery, and they travel through the cord and go through the dead battery, and eventually they go back to the other battery, right? So they have to come from somewhere, they have to go somewhere. If you do something wrong, or not wrong, if you break that, then the electrons don't flow, just like in an electrical circuit. Same thing here. Anyway, any questions about that before we finally get into the details of the third stage? All right, then. So again, third stage, electron transport chain. Happens in the mitochondria along with the citric acid cycle, and it produces ATP, the most ATP. So again, this is a whole downward trip for electrons. They're high energy when they begin, and by the end, they are low energy, right? They're just regular old electrons. So as I've already said, now you can see it writing, those electrons start from the glucose, right? And then the NAD plus strips them away. It takes them to the electron transport chain, where finally they are ultimately accepted by oxygen, and that forms a molecule of water. Very important for the exam right there. So many test questions. Like the one I already said, where do the electrons come from? The glucose. What's moving them? The NAD plus, right? It's the NAD plus that's stripping them away. Which molecule ultimately accepts them? Oxygen. And what molecule is formed when that happens? Water. You definitely need to know that. Electrons go from glucose to oxygen, forming water. Make sure you know that. That'll be important, because then you have to learn about photosynthesis. And photosynthesis is the opposite. That at least you can still associate those three things together. In photosynthesis, water gives up electrons, which forms oxygen, and those electrons that it gave up are used to make glucose. So it's completely the opposite. But for this exam, if you can just associate this. When we're talking about electrons, you associate glucose, oxygen, water. Always associate those three things. First, memorize that. Then memorize the order. If you can at least memorize that those three things go together when you get a question about where the electrons come from and where they go, then you're good. Because on the exam, if you guess this, but in the wrong order, if you have these three things on there for your answer, but they're in the wrong order, you'll at least get partial credit. Because they knew. They just had like if you say the oxygen gave up the electron, or the oxygen gave the electrons and glucose accepted them, forming water, well, at least they got the three parts. And just so you know, too, that's one of the few questions on the exam that will not be multiple choice. That'll be a fill in the blank. It'll say something like, in cellular respiration, electrons come from blank, and they're ultimately accepted by a molecule of blank, forming a molecule of blank. So that will be not a multiple choice question, but a fill in the blank. Anyway, any questions about this slide before we talk more in detail about the electron transport chain? This is still big picture stuff. And again, like I say, my recommendation for studying this entire exam is look at the big picture and once you have the big picture memorized, then start adding details. For example, let's see if someone knows the biggest picture question there is. What is the whole purpose of this whole thing? Citric acid cycle. What are we trying to make? I'm sorry. Good grief. Cellular respiration. This whole thing we've been talking about. What is the ultimate thing that we're trying to make? Yes, good. I'm so happy someone got it. Hopefully, too, everybody else got it, and you just were afraid to say it. Yes, ATP, right? If you don't know that, then forget all these other little details, right? Anyway, let's move forward. Now, let's do one more attendance word. I'm going to point at it. There it is. I'm not even going to say it. So if you're online, nice and relaxed and listening to the lecture with your eyes closed, which is okay, open your eyes and look what the arrow is pointing to, because that is the next attendance word. But again, there's more. This is just the overview. Now that we know that big picture, let's get into some more details about the electron transport chain. This is what it looks like. This is a picture not from your textbook. This is from your old textbook, but this is a great visualization of what we're saying. We've stripped away these high energy electrons from glucose and other stuff that we'll talk about later. Well, we know glucose, right? And then pyruvate, and then acetic acid, and then citric acid, so on and so forth. But anyway, we stripped away high energy electrons. Those electrons are brought to the electron transport chain where they continue this downward trip where they're releasing energy, right? And like I said, oxygen is the molecule that accepts those high energy electrons. And when it does, it forms a molecule of water. You'll notice, too, that this picture has more details than I've talked about because I'm not going to test you on those details. Yes, technically, NADH holds 2 electrons. I don't care about the numbers. Doesn't matter. Yes, technically, those 2 electrons, like if you were to balance the equation, would meet up with half of a molecule of O2, and that would also be combined with 2 protons or 2 hydrogen ions, however you want to say that, to form a molecule of water. You don't need to know that much detail. What you need to know is that these high energy electrons come from the glucose. They're stripped away. They go to the electron transport chain where they release energy. Oxygen ultimately accepts them, forming a molecule of water. So all those numbers don't matter. The hydrogen ions don't matter. I mean, to us, for testing purposes. But here's what I haven't told you yet. We're going to get a little bit deeper. So again, you study the big picture, then start adding in details. Memorize this before you move on to what I'm going to teach you next. Because what I haven't taught you yet is this process here. All I'm showing you here is that as these electrons go down the stairs, so to speak, they're giving off energy, and that energy is used to make ATP. But I haven't told you how. So that's what's coming next. So again, my recommendation is when you study, memorize this first. Once you have that, once you're like, I know that, then you can dive a little bit deeper and figure out how that works. This is what we're about to do. So any questions about this slide? All right. So there are proteins, or the proteins of the electron transport chain, or oxidative phosphorylation, whatever. They are built into the inner membrane of the mitochondria. This is going to be a horrible picture, but I've shown you the picture before. Wait, no, I'll show you next slide. I'll show you on the next slide. So I'm going to talk about it really quickly, then I'll show you, and then I'll see if you have any questions. These membranes are highly folded because that gives you a large surface area. That'll make sense when I show you. Let me put a big old X to this. So this is not important as far as the test is concerned, because I'm not going to ask you where you find the proteins of the electron transport chain. I'm not going to ask you if the inner membrane or the outer membrane is more folded or why it's folded. This is just for your own knowledge so you know how it works. So here we go. Let's look at it and talk about it. There we go. So again, there's 2 membranes. Unlike our cell, right? Our cell has the one membrane. Sure, it's a phospholipid double membrane, but it's still only the one membrane. Here we have a membrane inside of a membrane, if that makes sense. Think of like a cell being swallowed by another cell. As far as membranes are concerned, that's what we're looking at here. And you can see the outer one is nice and smooth, right? But the inner one's all wrinkly. Because imagine having a small garbage bag, like a Kroger bag, you know what I'm talking about? And then taking a big old garbage bag, like something from that trash can. Which one's bigger if I were to hold it? I know you can't see it. But if I had a Kroger one here and a regular garbage bag here, which one would be bigger? The Kroger one or the regular one? Regular one, right? So, but you still could get that regular garbage bag and crumple it up and put it in the Kroger bag, right? Kroger bag will be nice and full. The inner bag will be all crinkly, right? Because you have a big old bag crunched into a little bag, right? That's what this is. Technically, what you really have is a big old bag inside of a smaller bag. And that's important. And the last slide mentioned this, but I'm going to try to draw it. There's little proteins embedded here. And these are the proteins that are doing the work. These are the electron transport proteins. So if they're embedded in the membrane, obviously the more membrane you have, the more proteins you have. Does that make sense? The bigger the bag, so to speak, the more proteins you have. Therefore, the more effective this is. The whole idea here is to make ATP. And if these little red dots are the things making the ATP, we want more of them. So anyway, I'm not going to test you on that, but it's just a good example of form and function, right? That's why it has this shape. Another thing I'm not going to test you on, well, basically this whole slide. You don't need to know the difference between the inner membrane or the outer membrane. You don't need to know what the cristae is or the matrix or the space between the membranes. But there it is for you. Any questions about that slide? All right. So some more big concepts before we talk about specific concepts. Energy is stored by the electron transport, and it's something like the water behind a dam. So I don't know if you guys have ever seen a hydroelectric dam or any dam, right? But as you can imagine, on the other side of this dam is a whole lake, right? A big body of water, and it's just holding all that water back and has little areas where it lets the water through, right? Pretty simple concepts. In your mitochondria, it's a lot like that. Your inner membrane is a lot like that dam. And in between the inner membrane and the outer membrane is the water, so to speak, right? And it wants to come inside. That water, well, I'll go ahead and tell you it's not water. It's right here. Instead of having water, it's hydrogen ions. So if this dam were mitochondria, there'd be a bunch of hydrogen ions over there, and they would just naturally want to flow downhill. Make sense? If that was hydrogen ions instead of water, that's how a dam would work. You guys with me? So in this case, what you have is a bunch of hydrogen ions caught in between the membranes, and there's barely any hydrogen ions on the inside. So as we know from diffusion, we know that those hydrogen ions want to go inside, right? Because they're stuck. They're really highly concentrated in between these membranes. So they want to go inside where there's a lower concentration. That makes sense. Nice and simple concept. Stuff you're already very familiar with. Diffusion. Going from an area of high concentration to low concentration. So it's the same as this dam. You have all those hydrogen ions wanting to go inside, but there's these things keeping it. Keep that in mind and we'll talk about it. So the mitochondria have structures that act like those turbines. So just like in a hydroelectric dam, again, it opens up, it lets the water flow through, and the water just flows through naturally because that's what gravity does, right? And it goes and it turns the turbines, and that produces electricity. Well, it's similar here. Mitochondria are keeping those hydrogen ions dammed up. They want to come inside, but they're not letting it happen, right? But then these enzymes, and you do need to know the name of this enzyme, these ATP synthases are basically like the turbines. They're like the little holes in the dam. They let the hydrogen ions through, and they just flow through naturally, right? Because it's diffusion. They go from an area of high concentration to low concentration. And as they just naturally flow through, it really does turn. There's a thing that turns on that enzyme. It turns it, and that produces ATP. But most importantly for the exam anyway, what you need to know on this slide, test question is going to be, in the electron transport chain, which enzyme produces ATP? Lucky for you, it's in the name, ATP synthase, right? ATP synthase is the enzyme that produces ATP. So any questions about this slide before I give you a little visualization? All right. It's a great picture. Excuse me. This is also not in your textbook. This is from your old textbook. And this is a lot more complicated. This shows you, again, you're getting like the Cliff Notes version. Even 120, which is the 100 level majors version of this course. So it's this course, but it's for majors instead of non-biology majors. They would have to know this. They would need to know all these details. This protein has a name. This protein has a name. This protein has a name. That chemical reaction has a name. That chemical reaction has a name. They all have names. Everyone else needs to know them. You don't. Again, what you need to know is that these high energy electrons were stripped away from the first two stages. They were brought to the electron transport chain where they go from protein to protein to protein. And again, those proteins are embedded in the membrane. of the mitochondria, as you can see here, right? But here's what I haven't taught you yet. What I did say earlier was those electrons, they're high energy and they can't stay that way, and they're releasing their energy. So finally, we get to end this story, so to speak. I get to tell you the whole picture. As those electrons go from protein to protein, and as they release that energy is put to use. That energy is used to pump hydrogen ions against their concentration gradient. So remember in the last chapter when you learned or maybe it was this chapter, whatever, earlier when you learned about active transport, there you go, there's active transport. Active transport is when something goes against its concentration gradient, and it takes energy to do that. In this case, we're pumping hydrogen ions against their concentration gradient because it's really concentrated here. It's not very concentrated in here, so it takes energy to pump them in there. It's those electrons that give those energy. And again, as we've already said, here's the recap. Now we have all these hydrogen ions at a high concentration, and they just want to naturally diffuse back here where it's a low concentration, and they can't anywhere else except through the ATP synthase. So they all just flow through here. And as they do, that thing literally turns, not that you need to know that it turns, but anyway, as those hydrogen ions flow through the ATP synthase, it makes ATP. So again, a lot of test questions on this slide. What molecule is the ultimate acceptor of the electrons? Oxygen. What molecule is formed when oxygen accepts those electrons? Water. Which of the three stages makes the most ATP? Electron transport chain. Oh, here's one I haven't said yet as a test question, but I've explained it already. Which of the three stages pumps hydrogen ions against the concentration gradient? Electron transport chain. Which three stages uses hydrogen ions going down the concentration gradient to produce ATP? Electron transport chain. Actually, here's a hint. Any questions that you get on this exam, when we're talking about cellular respiration, anything that asks about hydrogen ions, you should think electron transport chain. Because remember, this is the only time I've ever talked about hydrogen ions in this discussion. We don't talk about them in glycolysis. We don't talk about them in citric acid cycle, but we do talk about them in electron transport chain. And I like this little picture. Again, this is from your old textbook. Any idea why there might be a skull and crossbones right there? Has nothing to do with pirates, I'll tell you that. Yeah, death. Exactly. Any guesses why? So there's a lot of poisons, and a lot of poisons, this is how they kill you. You can look that up for independent work too. A lot of times you'll take a poison, and if it kills you, well, there's a lot of different ones, but some of them, this is how they do it. They prevent oxygen from accepting those electrons. Therefore, the electrons quit flowing. Therefore, you quit making ATP. Therefore, you die. Also, that's how you die when you drown or get choked or whatever, right? If you don't have oxygen, that's the step. That's exactly where it kills you. Because once you get rid of that oxygen, it's like cutting the cord on the electrical cord. The electrons stop flowing, you quit making ATP, and you die. There are some exceptions to that, and we will talk about that. Good, this is good timing. Cellular respiration generates about 32 ATP per molecule of glucose. I've already told you that. You need to know it. I'm not big into numbers. That's a number you need to know. About 32. And this slide shows you where they all come from. Again, I know we're almost done. A little bit, a lot, right? That's what you need to know for the exam. And when we get back, we will talk about the exception. Because like I said, if you don't have oxygen, you die. There's an exception to that. We'll talk about it when we come back on Friday. And so I'll either see you on Friday or see you in lab today. Oh, sorry. The last word for attendance then. Nah, let's not do it. We're good.